Welcome to Microbial Metabolism. So this is chapter 8 in the Microbiology OpenStax textbook. And so metabolism in a nutshell is the sum of all of the chemical reactions that occur within body cells or within cells. And cells have to be able to synthesize parts, but they also have to be able to produce energy that will allow them to then synthesize those parts. And so this definition then is all the chemical reactions in an organism is their metabolism. They can be broken into two major reactions, either catabolic reactions, which break down molecules, or anabolic reactions, which build up molecules. So catabolism is the breakdown of large molecules into smaller ones, and in the process we release energy. Anabolic reactions then are the buildup of smaller molecules into a larger one, and we use energy. So catabolic reactions can drive anabolic reactions. A catabolic reaction is also considered an endergonic reaction. And an anabolic reaction is also considered an exergonic reaction. So the term endergonic means the product has more energy than the reactants had. And so you have to have an input of energy. In an exergonic reaction, the product has less energy than the reactants. And so we're releasing energy. So organisms are classified by how they gain their carbon as well as how they gain their energy. So we have autotrophic organisms that gain carbon by from inorganic carbon like carbon dioxide. We have heterotrophic organisms are going to gain their carbon by organic compounds like carbohydrates. Organisms that are classified via energy are either going to be classified as phototrophs, which obtain energy from sunlight, or chemotrophs, which gain energy from chemical compounds. So here we have these chemotrophic organisms. They can be chemoautotrophic or they can be chemoheterotrophic. So if you are a chemoautotroph, you gain your carbon source, that's the autotroph, by inorganic carbon, but your energy source is chemical, so an organic molecule. If you're a chemoheterotroph, you use organic and the chemical source of energy again. If you're a photoautotroph, then you use the photo, again, you're going to use light energy. And if you're an autotroph, again, inorganic carbon, heterotroph, organic carbon. And I gave you some examples of those different organisms over here. Two other terms that we often use in metabolic reactions are oxidation and reduction. So you've all learned in a chemistry class in the past, hopefully at least, that the term oxidation means loss of electrons. And the term reduction means gain of electrons. Oil rig is how a lot of people remember this. Oil, O, is oxidation. I is loss, L, okay? And then rig, reduction, is gain of those electrons, okay? But in biology, we add those hydrogen ions as well. So here we have an agent called a reducing agent that is going to give electrons and hydrogen to some other molecule or some other atom in this case. So here we have a reducing agent. It gives an electron and a hydrogen to this one. This is called an oxidizing agent because it's going to take those electrons and hydrogens. So the reducing agent becomes oxidized. The oxidizing agent becomes reduced. And those are coupled reactions. Anytime we oxidize one thing, we have to reduce something else. In metabolic pathways, we have to have special helper coenzymes. So coenzymes are helpers to different metabolic pathways. And in the metabolic pathways here, we're going to be talking about 3 coenzymes: nicotinamide adenine dinucleotide, flavin adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate. NAD and FAD are both part of the catabolic pathway of cell respiration, whereas NADP is part of the anabolic pathway of photosynthesis. And so while we oxidize certain molecules, we're going to reduce NAD, FAD, or NADP in these different metabolic reactions. And then in the end, they will become oxidized and go back and do their job again. And something else will get reduced. So ATP, adenosine triphosphate, is the energy currency that our cells use. ATP is considered a very high energy molecule. The energy is found between the 2nd and 3rd phosphate, and when we break that bond, we release the energy to do different activities. And when we do that, we produce ADP plus this inorganic phosphate. The last thing in this video I'm going to talk about are what enzymes are. Enzymes are organic molecules. Most of them are proteins, and their function is to catalyze a reaction. So a catalyst speeds up the rate of a reaction, and it does this by lowering the amount of energy that is needed for that reaction to occur. In our body, every chemical reaction that occurs uses enzymes. Metabolic reactions would still occur without enzymes, but they would not be fast enough to sustain life. So enzymes are pretty awesome. They're reusable. They are able to bind to only specific molecules, so they respond very easily to their substrate. And they can be used in very small amounts, so they can catalyze lots of reactions every single minute. So the substrate is what an enzyme binds to. And so here's the active site of an enzyme, and this is the substrate that will bind to that active. When a substrate and an enzyme bind, the enzyme actually changes in configuration just a little, and this is known as an induced fit. And so that change in configuration actually stresses the bonds in the substrate, and it'll either produce new bonds or it'll break different bonds. So we are either going to build something up or break something down. Sometimes enzymes need something to help the substrate to work with it. And so we have inorganic ions that are called cofactors, like metal ions, or we have organic molecules like coenzymes, and we talked about NAD, FAD, and NADP. Those are all coenzymes that help the enzyme function properly. Enzymes can be controlled through both competitive and non-competitive inhibition, and we can actually use this information to treat certain infections by stopping like bacterial enzymes, so that we then can stop the bacteria from being able to replicate. So competitive inhibition occurs when some non-substrate binds to the active site. And when it binds, it changes the shape, and the substrate now can no longer bind. Non-competitive or allosteric inhibition occurs when again another molecule binds to a region called the allosteric site. When it binds, it changes the shape of the active site, and again the substrate cannot bind. So feedback inhibition occurs in metabolic pathways when one of the products that are produced stops the metabolic pathway. And so here we have enzyme 1. Enzyme 1 binds to a substrate. This produces a product. This product is then the substrate for the next enzyme. Here we have another substrate binding to an enzyme, and this produces a new product. This product is now the substrate for the third enzyme. And so when this enzyme binds to its substrate, then this product is produced. And here we have this product. This product can then bind to the allosteric region. And when it does that, it changes the shape of the active site of that enzyme number 1. And by doing that, it prevents synthesis of some product. So this is really important because if we have a lot of some product, we don't need to produce any more of that product. So that actually benefits us. It basically reduces the amount of energy we're expending producing certain products. I'm going to stop here, and then in the next video, I'll talk about aerobic respiration. Bye.